Hello, everybody. It's a great pleasure to start this webinar on COVID-19 immunity with my co-chair, John Friedland from Imperial, myself, Annelies Zinkernagel from the University Hospital of Zurich in Switzerland. We're infectious disease specialists co-chairing this webinar on COVID-19 immunity. We have three distinguished speakers, each with 30-minute time slots for the symposium as well as the discussion. Please send us your questions via the Zoom chat that we can also address those after the talks. And with no further ado, I would like to start introducing our first speaker, Andrew Pollard, who is a professor of pediatric infection and immunity at the University of Oxford in England. He obtained his medical degree at the St. Bartholomew's Hospital Medical School at the University of London. He then trained in pediatric infectious diseases and has been developing various programs, including the design, development, and clinical evaluation of vaccines of bacterial pathogens, but as well as of viral pathogens, one of which we're talking today about COVID. So he's the chief investigator leading the development of the AstraZeneca vaccine against SARS-CoV-2, the COVID-19, which he will be talking about. He also received numerous awards over the time, one of them from the European Society of Pediatric Infectious Diseases, as well as the Rosen von Rosenstein Medal in 2019. He was awarded the Swedish Pediatric Society Award and many others, and I'm looking forward to your presentation. The board or the Zoom is yours.
Thank you very much, and thank you for inviting me to speak in the symposium. And I'll talk to you about the development of the Oxford vaccine. Can you see the slide on full screen?
Yes.
Perfect.
It's not full screen, it's the representation mode. So we always see the next slide following.
I think that's what, no, I think you are seeing it full screen, Annelies. I think that's the way this is set up, is it not?
Do you all see the full screen, or?
I'll try sharing again. Is that projecting on full screen?
Yes, now it's correct.
Yes.
Looks great. Perfect.
Okay, great. Slightly confusing setup on the system. Okay, so I'm going to talk about the development of the Oxford vaccine, and it's actually a great moment to be able to speak about it because just over the last 10 days or so, really we've started to see the rollout of the vaccine through COVAX to many low-income countries around the world. And when we started out a year ago, that was a major part of our mission, was to be able to develop a vaccine which could be accessible equitably everywhere around the world. The first doses arriving in Ghana and now into many countries over the course of that last couple of weeks. So very, very exciting for me that we've reached this point. And this is just from the end of last week, where COVAX had already shipped millions of doses to 19 countries. Of course, where we are today is with a huge amount of the virus still spreading in many countries around the world. And particularly concerning is some of these increases that are being seen in some parts of the world, particularly in Latin America at the moment, of the increased transmission, possibly related to new variants, with huge numbers of cases reported from around the world, but underreporting in many regions because of lack of access to the testing to demonstrate the cases. An astonishing mortality rate already reported, and again, those figures likely to be an underestimate of the total number of deaths caused by this pandemic virus. And the reporting very much dominated by the United States and Brazil, as you can see on this slide, with very high numbers of deaths already. And of course, there is a positive story in that there are 79 vaccines in clinical development already authorized at this moment, and an astonishing 182 vaccines that are waiting in the wings in preclinical development, which hopefully we'll have access to soon. There are 7 billion people on the planet, and although around 250 million doses of different vaccines have now been administered around the world, we have a long way to go to protect all of those who are vulnerable everywhere. You can see here on this slide in vaccination doses administered per 100 people that some countries have got very good, well-organized systems, particularly, as you can see here, Israel, the United Arab Emirates, which have really got ahead of the vaccination strategies to give doses to those across their populations. And we have a number of different tools, but in the end, they're all about the generation of proteins from the coronavirus, and particularly the spike protein, which is the basis of most of the vaccines that have been developed, with the immune responses generated to spike proteins. So some, obviously, the protein vaccines involve the administration of spike protein itself. Many of the other vaccines harness this cellular machinery of the muscle or the connective tissues after vaccination to allow the expression of spike protein, whether it's by RNA, DNA, or viral vectors. And the viral vector vaccines are the ones that we've been developing here in Oxford. And the coronavirus is decorated on its surface by this spike protein, which binds to the ACE2 receptor. And the genome was available back in January, but at that time, it was very uncertain how important this virus was going to be. And in fact, this New York Times article points out that this new virus has surfaced in Wuhan. The region's on alert, but it doesn't appear to spread between humans. And how wrong this article has turned out to be with this global spread of the virus. But at that time, my colleague Sarah Gilbert started working on putting this gene insert into our vaccine platform to be ready to deploy if it was needed. Normal development it takes between 5 and 15 years. So at the beginning of this program, it looked like an almost insurmountable problem to be able to get through all of these stages of development to authorize a product during the course of the last year. But we did have some real advantages. One was that we had the genetic sequence, because science has brought us to the point where genomes can be emailed around the world at incredibly high speeds. We also had the experience of 2 previous outbreaks of coronaviruses, the SARS coronavirus outbreak in 2002 and MERS about 9 years ago. With these 2 coronaviruses didn't spread well between humans, and they were halted, the outbreaks were halted by case isolation. But now these really did make us very concerned because of the high mortality. About 11% of those who developed SARS died, around 1/3 of those who developed MERS died. And that meant there was a big drive around vaccine development to make sure that we had vaccines tested and we understood more about their use before this pandemic occurred. And so there'd been a lot of animal testing. We had the first studies of the MERS vaccines in humans were already underway when this pandemic began just over a year ago. And so with the vaccine platform that we have available here in Oxford, a chimpanzee adenovirus, which is non-replicating, so it can't cause infections in humans. It doesn't, it's perfectly safe in the immunocompromised individual. And with the knowledge that we had the genome from the virus and could put the spike protein gene into our viral vector, we were then in a position where we could immunize individuals with the adenovirus, which would then unpack, not cause any problems because it can't replicate, but lead to the expression of the spike protein in the cells in the volunteer to then generate immune responses against spike protein. We got these studies underway on the 23rd of April last year with 1,000 individuals in this first study, in an attempt to understand the safety and the immunogenicity of the vaccine. And we found that very rapidly that we were able to generate these data. We had hoped that with this large study we might get some efficacy data, because the predictions were that we'd be vaccinating at the tail end of the first wave of disease in the UK. But the lockdown was so effective that actually there were very few cases that occurred during the time that we were recruiting in this phase 1 trial. But we were able to demonstrate strong immune responses to one dose of vaccine, which were boosted with a second dose. And these were neutralizing antibody responses, but also T-cell responses peaking 2 weeks after the first dose, and showing strong both cellular and humoral immunity to provide protection against the coronavirus. Importantly, the T-cell responses were Th1 biased, important because this is thought to be protective against the potential risk of enhanced disease. And so these were the right sort of immune responses which regulators were hoping to see from these vaccines. Having started enrollment in younger adults, a key question was about immune responses in older adults, where we know with most vaccines we see much poorer immune response. And so we enrolled groups of older adults, those over 56, those over 70, to evaluate the immune responses in these age groups. We saw very similar T-cell responses as with younger adults. And here's some of the neutralizing antibody data showing that whether you're over 70 here in gold, in green, the younger adults, or the group in the middle in purple, all of them made very similar immune responses to the vaccine, and that 2 doses gave higher responses than a single dose. And for that reason, we progressed with our development program as a 2-dose schedule. As we moved into the phase 3 studies with around 24,000 adults, we took on sites in Brazil and in South Africa to support the development effort and to make sure that we had sufficient individuals enrolled over the course of the year. And those sites are based in Brazil and in South Africa and here in the UK. And a partnership with AstraZeneca we made in April, because they shared our vision of the vaccine being a not-for-profit vaccine, so that we could make sure that there was equitable access, whether you're in a wealthy country or a poor country, to the vaccine. And they have set up trials in the United States of about 30,000 people, and smaller immunogenicity trials in Russia and in Japan. And a further partnership with Serum Institute of India, the largest vaccine manufacturer in the world, who have run trials in India, so that we were able to cover all geographies around the world in our trials, and importantly, to evaluate the vaccine in different social settings and ethnicities, as well as different geographic regions, which is very important for building confidence in the vaccine as we move forwards. So by the end of November, we'd enrolled over 23,000 volunteers, collected around 100,000 blood samples, and had 500,000 pages of data to go through and clean, ready for the interim analysis, which was presented at the end of November. And critically, there were no hospitalization, severe cases or deaths in anyone who'd received at least 1 dose of the vaccine. And we saw high efficacy, 70% efficacy against disease, including the mildest disease in these trials. So in the UK, with the data submitted from these trials, we received regulatory approval for an emergency use authorization at the end of December. But the trials hadn't finished, and so we continued to generate more data during the course of the year, particularly as new waves of disease started in various parts of the world. And we continued to see this high protection against hospitalization, that once the immune response is kicked in after the first dose, that no one in the vaccinated arm is hospitalized. And importantly, and the regulators and the policy bodies have looked at these data to generate the current policy here in the UK, is that after the first dose of vaccine, we see very high levels of protection that are not decreasing at all during that 3-month period between the first and second dose, which is currently recommended. So the first dose is given high levels of protection, and that persists right up until the second dose is given 3 months later. And that's supported by the antibody data, which And this is over a 6-month period after a single dose. But even if we look here where the second dose is given, there's almost no decline in the antibody over that period of time. And this has really been borne out now in the real world effectiveness studies here in the UK, where after a single dose, even in the oldest adults, there's high levels of protection against hospitalization in those over 80 or over 70 in these 3 different studies, very similar to the protective efficacy seen after the Pfizer vaccine in these same studies. And I think this is really good news that right from vaccinating populations with a single dose, and if you start with 1 dose and give the second one 3 months later, you've got a lot more people vaccinated in those first 3 months. So what about after the second dose? And with the longer dosing interval, we see higher efficacy after the second dose, which you can see on this slide increases gradually from the second dose being given 28 days up as the dosing intervals extended, as we see with other vaccines, that if you give time for the immune response to mature, the booster response is better after the second dose. So the development of the vaccine has taken place now over the course of the past year. From the first sequence being available in January, it's taken 11 months to the first authorization. And from the first needle given in a trial, it's only 8 months of vaccine development. So rather shorter than the 5 to 15 years that we normally see in vaccine development. And this has been possible because of the focus of all of the research teams involved around the world on this vaccine development program, and of course the support from funders and from the regulators to make sure the process could move as quickly as possible to help all of us be protected in the pandemic. From that first approval, we had the first doses given in the national rollout here in January, and here I am receiving my dose. And the European Medicines Agency gave their approval for all ages at the end of January. And importantly, this then fed into the World Health Organization's policy decisions and the pre-qualification of the vaccine by the World Health Organization, so that doses made in India, Covishield, and also doses made in Korea could both be used into the COVAX facility, so that by the end of February, doses started to be shipped to vulnerable populations everywhere. So we have proven efficacy of the vaccine in trials. The vaccine is now authorized for use in many countries around the world, and we have global rollout underway, and increasing evidence of strong impact of the vaccine across all ages in preventing severe disease, hospitalization, and death. And of course, it's only when we get to a point where our hospitals are no longer filling up with patients with COVID that this pandemic ends. And I think increasing evidence that these vaccines work and are saving lives today. I'll just end by thanking all of the team in Oxford who have been working on this with me, our investigators in Brazil and South Africa and across the UK, and all the trial volunteers who, particularly when we think back to a year ago when we were enrolling them, and we had to say quite honestly to them, we don't know if these vaccines will work, we don't know if they're safe, but we do want you to take part in these trials because if they do work, they're able to have a huge impact on humanity if we can roll them out quickly enough. Thank you.
Thank you very much, Andy. It was a great talk. Please do submit your questions via the chat. We have had a couple of questions. And there's one question, or a couple of questions relating really to what is the reaction to the adenovirus and does that impact in any way on our immune responses?
That's a really good question. So we have looked at whether the neutralizing antibody that's induced by vaccination has an impact on the response to further doses of the vaccine. And so far, we've seen no relationship between neutralizing antibody directed against the adenovirus and any change in the response to spike protein. and whether that's the small number of people who have some pre-existing antibody or those who develop antibody with the first dose. There doesn't seem to be a relationship. Now, of course, that doesn't exclude the possibility that there are other mechanisms that inhibit responses. But we do see a boost with the second dose, and as I say, we can't find with neutralizing antibody relationship between the two.
Thank you very much. There's also a question being asked about potential viral integration into the genome. Would you like to comment on that? I don't think that is a big problem. But.
No, I mean, the adenoviruses that we all get normally as upper respiratory infections don't integrate into the genome. This virus is even replication incompetent. There's even less chance of that, even hypothetically, that it could do that. And these DNA viruses don't have the necessary machinery to integrate. So that doesn't seem to be a problem. And of course, it is studied in the laboratory to see whether that can happen, and we haven't seen evidence of it.
You'll undoubtedly be aware there has been various issues about the licensing of the vaccine in the over 65s, and I think it's not suggested in the chat that it's not approved in Canada yet in that age group. Would you just like to make a comment? You did touch on this actually in your talk about the age specificity, the range of age specificities, maybe broaden the question a little bit.
Yes, well, so the regulators who look at the safety, the efficacy, and the quality of the vaccine have everywhere have approved the vaccine for use in all ages, and the MHRA did, the European Medicines Agency, and actually Health Canada as well, have all approved use for all ages. And that's based on the data that show very good efficacy and good responses in younger adults, and in the older adults, very similar immune responses. And the efficacy data that there are also support the efficacy in older adults. The difference is that because we ran the trials in older adults starting a bit later, there's been less time to accumulate data. And also the older adults are a bit more cautious. So they have actually had fewer cases of disease in the trials. So we have less evidence. So statistically, less certainty in older adults. But all the data that we do have pointed in the same direction. And so the many recommending bodies around the world have looked at the data and taken the decision that it works in young adults, it works in 56 to 69, and it works in the over 70s in immune responses, and the evidence is all in the right direction. But some policy bodies look at the data and their rules require them to have more evidence of efficacy. And I think the problem hasn't been that scientific process, because every policy body has different set of rules to work by. There's a problem around communications. It's really important to communicate the difference between the level of evidence required for a policy body's decision and absence of evidence, which is one of the wrong messages that has been put out. Sorry, I mean the lack of protection, which is the message put out, which is incorrect.
We've got a couple more questions. I'm just going to ask you sort of another question, because as you're a pediatrician by background, is what do you think of the role of vaccination in the pediatric age group, where obviously I know there are ongoing trials looking at this, but I wonder what you know, casting your mind forward.
Well, I think there's several issues for children. First of all, there's a real issue for some children at this moment, particularly those slightly older children with neurodisability, where we do see higher rates of severe disease. And I think those are already in many countries being targeted for vaccination. There's, I guess, the second question, which is not yet resolved, is the extent to which the post-COVID inflammatory syndrome, PIMS-TS, is a widespread problem in children. We are seeing many cases around the world, and that may in itself reach a point where it justifies vaccination. I think we need to understand more about that. So there could be some direct benefits of vaccinating children, but most of the discussion really is about vaccinating children to reduce transmission and perhaps protect older adults. And that's a question which I think we can say something about today, which is the evidence from the trials is that there is a substantial impact on transmission of all of the vaccines where we have data. And with our vaccine, we did swab everyone in the UK every week. So we know about symptomatic and asymptomatic infection. And we see after the first dose about a 2/3 reduction in PCR positivity, and therefore we don't think those people can transmit. So there is an argument today, looking backwards, to say that it might be reasonable to vaccinate children to protect them, but also to reduce transmission. The difficulty is looking forwards, where the nature of RNA viruses is that they'll throw up new variants and transmission will continue for the decades ahead. I don't think SARS-CoV-2 is going to suddenly disappear from the planet. And for that reason, we've got to perhaps think about vaccinating children if we can predict which variant we're going to be dealing with and whether it's appropriate to be giving a vaccine that might need updating every year to all children. And I just don't think with this moment, we have a really clear view on exactly what comes next with this pandemic.
That's really helpful, thank you. And you answered a question also about transmission, so that's great. I think just coming on to sort of coming off what you just said then, what do you feel about this vaccine in the context of the new variants that are arising in different parts of the world, which are obviously all, well, not all, but there's a range of different variants with different susceptibilities and different responses to different vaccines?
Well, I think there's two aspects to this. The really important thing that we need to do that ends the pandemic is to have vaccination that stops people going into hospital and dying. And there is a suggestion from the data that are emerging that there may be a difference in the level of immunity, or maybe the type of immunity needed to stop lower respiratory tract infection from the immunity you need to stop transmission. The virus is changing to allow itself to survive in human populations, particularly those where there's immunity building through infection or through vaccination. And that, I think the feature of the virus in the future will be that it mutates to allow ongoing transmission. And I think the question that we have to address is whether the current generation of vaccines is going to be sufficient to still protect the lung in those individuals despite ongoing transmission. There's some hints to me which suggest that that might be the case, but we really need more data to understand that. And in the meantime, we're all working on updating the vaccines to be ready in case that's not the case.
Yes, thanks very much. That's very interesting. So I think we've got time for one last question, just about a minute or so. Someone's written that there's quite a few people get quite a strong immune response to the vaccine with, marked fever, malaise and headache after the first dose and the second dose. Do you know why this might be compared to obviously many other vaccines which don't seem to elicit quite such thing?
Yeah, I mean, so the driver of the immediate reactions that happen in the first 1 or 2 days after vaccination are innate immune responses. So there's an inflammatory innate immune response going on in that period. And the spectrum of symptoms is very similar, whether you have a Pfizer vaccine or a Moderna vaccine or an Oxford AstraZeneca vaccine. What we see is with the viral vector vaccines that those side effects that happen in the first couple of days are much more profound with the first dose and are almost non-existent with the second dose. Whereas with the RNA vaccines, it seems to be the other way around, where the reactions are a bit milder with the first dose and are greater with the second dose. And so I think the full understanding of what's driving those differences, I don't have at the moment, but it's something which certainly needs more evaluation. The other thing which is really important is age. And it's absolutely clear that reactions are much more common in younger adults than they are in older adults. So here in the UK, where the rollout of the program has focused on the oldest adults, we've seen much lower rates of reaction than in some other countries where the rollout has focused on younger adults who react more. And of course, there's also differences in the rollout of the two different vaccines. So in many countries, Pfizer has been used for the older adults and the Oxford AstraZeneca for younger adults. So we are seeing differences, but some of that will be related to age. And so I think it's actually at this moment quite difficult to do the comparisons in the way that you're suggesting. But I think that these are important things, and the key is communication. These are expected reactions that people need to know about. They need to know that they pass and that they should take paracetamol if they feel unwell. And some people feel pretty ropey for a day or two after they've been vaccinated.
Thank you very much, Andy. I think I we're going to have to bring it to a halt there. I mean, we've had lots of questions. I'd like to thank the audience for putting forward lots of questions. I'm sorry we haven't managed to get quite through all of them, but we got through quite a lot of them. It was a great talk, really enjoyed that. I know you're really busy today, so I really appreciate you coming along and participating in this seminar, and I'm sure the audience do too. This is the virtual clap, and
Thank you.
and we will have to pass on to our next speaker now. So thank you very much indeed. Our next speaker is Steve Pascalo, and he is an immunologist who originally trained in the Institut Pasteur in Paris, and he has worked in a number of environments, both the academic environment and the commercial environment, and he is indeed a founder of a drug company, but currently he is an academic in Zurich, and he's going to talk to us today on the mRNA vaccines and this rapid adoption of a novel technology. So, Steve, it's over to you. Thank you.
Thank you. So,
just need to put it on full screen. Perfect.
thank you for your invitation. I'm happy to present the data on mRNA vaccine, a little bit of history, and I changed a little bit my title. I added synthetic mRNA vaccines. I will come to this point in the next two slides. For my background, indeed, I had always an academic and industry parallel career in the University of Tübingen, and I co-founded CureVac in 2000, and I left the company in 2006 to be then in the University Hospital of Zurich, the University of Zurich, and also I founded here another company called Mischer Pharma in the name of Friedrich Mischer, the Swiss person who discovered nucleic acids in the 19th century. And I created in 2016 the messenger RNA platform at the University of Zurich. Let me start by having a look at the existing vaccines. It's a non-exhaustive list here, but you can separate the vaccines being given subcutaneously and the vaccine being given intramuscularly. All the dead vaccines, so inactivated viruses or subunit vaccines, or most of them are given intramuscularly, most of the time with adjuvants. The live vaccines here in the top, they're given subcutaneously. And they work because they are live, because they infect our cells. In that sense, they are already DNA or RNA vaccines. So vaccines like measles, rubella, mumps, yellow fever, they are mRNA viruses. When attenuated in the vaccine, they infect the cells after subcutaneous injection. They give their mRNA to the cells of the patient, of the individual, and the individual is making the proteins of the virus, is stimulating his own immune system and triggers an immune response. So I want to highlight that we have already done mRNA vaccines. It's just natural mRNA vaccines, mRNA produced by the cells that produce the vaccines. And now we just switch to a synthetic mRNA vaccine. But the principle of giving foreign mRNA to our body as a vaccine principle is already used since more than 50 years, especially with the yellow fever virus, which is old. I highlight it even more here. You see some representations of mumps or measles or rubella viruses. They all are RNA viruses, so they contain RNA, they contain many proteins, they contain lipid bilayer, they contain surface proteins. So, and this is a vaccine. If you think about it, it's a complex mRNA vaccine containing not only a mRNA, but many proteins and lipids. That's the same for the three viruses. Here is a synthetic or IVT for in vitro transcribed mRNA vaccine. It's more simple. It contains one mRNA, while the other viruses contain several mRNA coding for several proteins. Here in the IVT mRNA vaccine, we have one mRNA coding for one protein and a simple lipid bilayer without additional proteins there. And this is usually 4 different lipids mixed together to do a liposome. So the synthetic mRNA vaccine is a simplified version of the previously made mRNA vaccine based on viruses. Going to SARS-CoV-2, We have now in the world 12 vaccines that are approved. And so one is protein vaccine in one country, but a new protein vaccine from Novavax should soon be approved. 2 mRNA vaccines are approved, and a few non-replicating viral vectors, as we have seen, adenovirus-based AstraZeneca, which is the most frequently used in 74 countries, it has been approved, and a few other vaccines based on inactivated viruses. So we have 4 platforms: protein, mRNA, replicating recombinant, non-replicating recombinant viruses, and inactivated viruses. One should highlight that they don't code for the same spike. The mRNA vaccines, they code for a spike in a pre-fusion conformation. It has 2 proline substitution in the sequence, which maintain the spike protein in a pre-fusion conformation, which is not the case for the AstraZeneca vaccine. The recombinant adenovirus vaccine from AstraZeneca codes for a wild-type spike. It's not stabilized in the pre-fusion conformation. This could also indicate that the mRNA vaccine and the AstraZeneca vaccine, for example, they may trigger different type of antibodies, eventually recognizing different type of variants. I'm listing here in a bit naive way the different advantage of the different platforms of doing vaccines: peptide, plasmid, recombinant viral vectors, mRNA, proteins, inactivated virus or attenuated viruses. Here in blue are the ones available in the world or validated in the world for SARS-CoV-2. You see, I will not go in the detail in the different advantage, but what I want to highlight is that mRNA vaccines are easy to design just from the sequence. The upscaling is easy, as I will show in the next slide. Once you can produce mRNA for one disease, you can easily use your pre-existing GMP facility for another disease, which is not always the case when you use recombinant viruses, because you have to redo a virus and to reestablish the virus replication in vitro and ways to produce it optimally. And very importantly is the theoretical safety. Of course, all approved vaccines have shown to be safe, and in that sense, they should be given and they save lives. Now here, I want to speak about theoretical safety, because this is what have driven me to develop the mRNA vaccines. So mRNA, as you know, once it's injected, it will quickly be degraded and eliminated naturally, which is not the case for recombinant DNA viruses. And the whole development of mRNA vaccines since 1998 is, for me, this aspect of safety. Of course, the mRNA vaccines were not so efficacious 20 years ago compared to DNA viruses, but the aspect of safety, of the fact that what we inject is quickly degraded and does not leave any trace, was a big advantage, which is one of the reasons why we continued on the mRNA vaccines. So to focus on mRNA now, I want to remind you what is a mRNA. So here's a cell, here's a nucleus. The DNA is transcribed into pre-messenger mRNA, and then this pre-messenger mRNA is capped at the 5', which is the structure depicted here, polyadenylated at the 3'. It's also a little bit modified inside. There are some methylations and some editing events happening in the mRNA, and this mature mRNA codes for the protein in the cytosol. The advantage, as I said before, is that this mature mRNA is transient, and that's what we use in this IVT mRNA vaccine, is a mature capped polyadenylated, eventually modified mRNA. It's possible to produce this mature mRNA in vitro. It's quite simple, as I will show on the next slide, but the production can be done in 2 ways. Either you use the 4 canonical nucleotide, HCGU, and then you produce non-modified mRNA. And this non-modified mRNA can trigger toll-like receptors. So our immune cells have toll-like receptors, which are sensing in the endosomes the presence of nucleic acids, and toll-like receptor 7, toll-like receptor 3, toll-like receptor 8 are recognizing RNA. And when you inject non-modified RNA, this pathway is triggered and you get inflammation, which is the natural adjuvant effect of the injected mRNA. That's why all over the world, in all companies, mostly for vaccination, it's non-modified mRNA which is used, because then you get advantage of the adjuvant effect of the non-modified mRNA in the vaccine. So all over the world, we develop anti-cancer, antivirus, anti-allergy mRNA vaccines based on non-modified mRNA. Most of the mRNA vaccines in development against cancer at BioNTech, for example, are non-modified mRNA vaccine. Now there is a possibility to make modified mRNA, which, for example, contains pseudouridine instead of uridine. And this modification allows the mRNA not to be recognized anymore by the endogenous toll-like receptors. So this modification that has been published in 2005 was coined to be used for gene therapy. So the idea here was to inject mRNA and not to give an immune response, so the opposite of vaccine. The whole principle of modified mRNA was gene therapy, expression of therapeutic proteins like erythropoietin or antibodies. So the idea is that you can inject this mRNA, not get an immune response, so that you can do in principle gene therapy. Only recently it was shown that although it's counterintuitive and it was not the point of the modification, but when you inject intramuscular this mRNA, it can be used for vaccination, probably because the adjuvant effect is there is still a little bit of adjuvant effect in this modified mRNA, or because of the lipid. It's not clear yet what's the adjuvant effect in the modified mRNA vaccines, but most of the time for vaccination, it's non-modified. The production of mRNA is simple and straightforward. You need only a few reagents. You need a plasmid DNA containing the gene of interest here in red, preceded by a promoter recognized by your RNA polymerase. You cut the plasmid so that it's linear here, and then in vitro, you add RNA polymerase and the nucleotides and the cap analog so that you generate mRNA. And from one molecule of DNA, you get over 1,000 molecules of mRNA. So the process is very efficacious, it's robust, it works independently of the sequence of the gene that is located here. So you can produce mRNA coding for any protein of interest using the same method, it always works. After the transcription, you put the DNase to degrade the DNA. And after that, you can purify the mRNA by lithium chloride precipitation to go fast, but you can also do, of course, some more advanced purification processes to have cleaner mRNA. So all these 4 steps are all robust and fast. So you can produce quickly mRNA coding for any gene of interest. That's one of the advantage of the mRNA-based vaccines. A few facts on synthetic mRNA, because we hear some fake news or some wrong information. First, one has to say that mRNA is very stable in the absence of RNAs. But DNA is stable in the absence of DNAs. Proteins are stable in the absence of protease. It's the same for mRNA. And actually, mRNA is indeed very stable. I will not go into the detail, but the EU has given a 2 million prize in 2014 to CureVac for vaccine transportation and storage where cold chain cannot be guaranteed. Because mRNA, free of RNAs, is very stable. You can store it at room temperature. If you lyophilize it, then you can store it for a very long time. And on top of it, I would even say, to be provocative, that mRNA is the most stable biological molecule. It's the only one you can heat to 95 degrees. If you make DNA vaccine, protein vaccines, or viruses vaccine, you cannot heat them up to 95 degrees. They would be completely deactivated. The mRNA, at the end of the GMP production, I had a step of 95 degrees heating. So because that's possible with mRNA, and it's not possible with other formats of biological molecules. So actually, mRNA is the most stable of the biological molecules. Now, the RNA liposome formulation may not be stable. So that's why the vaccines have to be stored frozen most of the time from BioNTech and from Moderna. And this is not because of the mRNA, it's because of the liposome, because the liposome at room temperature may aggregate, change size or form over time, and may be damaged by freeze thaw. So the liposome is the reason of the storage at low temperature of the vaccine, not the mRNA. Now one should also know, because people speak about a new technology, but RNA in liposome is already approved as a drug. There is a drug called Onpattro. It has been approved in 2018. It's the siRNA, so it's not mRNA, it's siRNA. And it's given at up to 30 milligram intravenous every 3 weeks. So we already have on the market an RNA drug in liposome. It's of course not mRNA, but just for people to know that we already, since many years, inject quite high amount of mRNA, or of RNA, sorry, in patients. The production that I showed you on the page before, it's interesting to note that it's vegan. The whole production uses products from the chemistry or from bacteria. So this is an advantage for certain populations who eventually want vegan products for style of life or religion reasons. And then I will just highlight that in general, in as few as 6 liters of in vitro transcription, you can make 1 million of mRNA vaccine doses. While when you do viruses, recombinant viruses, for example, usually depending on the virus, for 1 million of those, you need 5,000 liters. So you can see the scale and the equipment that are needed to produce 1 million of doses of a vaccine are much bigger when you use a virus, for example, or it would be the same for bacteria, than when you use mRNA. Some timelines of the mRNA vaccines. So the fact of using in vitro transcribed mRNA to express proteins in vivo was disclosed in 1990. And the first mRNA vaccine was actually disclosed in 1993. So it's a French team who has packaged mRNA coding for influenza protein in a liposome and could show in mice that this mRNA liposome formulation triggers an immune response. There was a similar work published 2 years later with naked mRNA coding for tumor antigens. And in 2000, Ingmar has published another mRNA vaccine formulation, which was made in the department of Professor Ramensey, who had the idea in 1996 to use mRNA for vaccination. And he gave the project to Ingmar, who was a student of Professor Gunther Jung. And in 2000, all of us with Florian von der Mulve, we created CureVac because we didn't want to do only publications. We believed in this technology being safe and efficacious, and we had to create a company in order to go to clinical studies. And one of the first challenge of the company was to manufacture the mRNA because it was not available anywhere in the world. So we had to implement our own pharmaceutical mRNA production, and this was validated in 2006. Earlier, we could do some individualized vaccines with mRNA and also injections in healthy volunteers, myself actually. And then in 2008, we published the first mRNA vaccine clinical study, which has been overlooked, but it was published in 2008. And there was also at this year the funding of BioNTech and later the funding of Moderna. Here is the experiment of showing the first injection of mRNA in humans. This is in me, don't be scared. We didn't ask anyone else to do it. I got injected with mRNA intradermally, the mRNA coding for luciferase. And 1 day later, under local anesthesia, we did punch biopsies to measure the luciferase expression. As you see here, in these punch biopsies, there was luciferase expression, showing that in the human skin, we have the same data than in the mouse skin. So when we inject mRNA, we see mRNA expression, but the mRNA is translated into proteins. So for the SARS-CoV-2 ongoing and approved studies, so most advanced are the 3 companies, Moderna, BioNTech with Pfizer, and CureVac. So the first company who has started to inject healthy volunteers was Moderna, with the first healthy volunteer injected on March 16 with the mRNA coding for the prefusion spike protein. There were 3 doses used in the phase 1. And for BioNTech, the first volunteer was injected 1 month later. But BioNTech had not only 1 vaccine, BioNTech had 4 vaccines, including mRNA coding for the prefusion protein, mRNA either modified or non-modified, while Moderna had only mRNA modified and CureVac has only mRNA non-modified for the vaccine. But so this study started in April, and the dose range in phase 1 was really broad, from 1 microgram to 100 microgram in different volunteers. And finally, CureVac has done a dose range between 2 microgram and 12 micrograms, and is now going into validation studies with 12 micrograms. From these 3 companies, 2 have been approved, the 100 microgram vaccine from Moderna and the 30 microgram vaccine dose from Pfizer-BioNTech. I will just show you here one example of what has published BioNTech from the phase 1 study. We focus here on the blue color. This is the mRNA coding for the full length spike. The red was a shorter version of the mRNA. And here you see placebo volunteers who receive placebo. This is the neutralization titer. So in placebo people, you don't have any neutralization titers against SARS-CoV-2. Here are the sera of patients who have recovered from COVID. You see that you have neutralization titers in all of them. And here in blue are the healthy volunteers who have received different doses of the mRNA vaccine coding for the full-length spike. These volunteers are older volunteers, and here are younger volunteers. You see that after the second injection, so at day 28, the second injection being given at day 21, in all healthy volunteers, whether young or older, and whether you use 10, 20, or 30 micrograms, all volunteers had, after the second dose of the mRNA vaccine, neutralizing titers similar to the one of the people who have recovered from the COVID, showing the efficacy of the mRNA vaccine at this very low dose of 30 micrograms. So I'm finished. I'm looking forward for your questions, and thank you for your attention.
Many thanks, Steve, for this wonderful presentation on the history as well as the current developments. We're extremely thankful that we have all of these vaccines. And with that, I would like to go to the questions we've received so far. One question is, can you explain how come some vaccines give higher antibody titers than the infection per se? I think in the last slide you showed that we're happy that we get similar titers, so I'm not quite sure where this question is going, but would you want to answer that?
I think it's wonderful that the mRNA vaccine and some other vaccines could give titers which are even higher than the one required for the virus to be cleared from the organism. I think one should keep in mind that most of the viruses, and it's the case for SARS-CoV-2, have ways to avoid the immune system and to downregulate the immune response. So it could be that the immune response given by the virus is kind of underestimation of indeed the immune response which can be given by a vaccine. The vaccine is only made to trigger an immune response. The virus has mechanism to avoid the immune response. So I think that's why the vaccine could give even higher immune responses than a virus.
Many thanks. Next question is, why are there such striking differences in mRNA amounts in the different vaccine formulations of the different manufacturers? What are the explanations for this?
I think all of this, so the three companies could have had, for example, a vaccine with 30 micrograms. So I think at the end, the Moderna has used 100 micrograms, but in the phase 1 with 25 micrograms, they had good results. So I think they could also have used 25 micrograms in their phase 3. And BioNTech has used 30 micrograms, but when you look in the clinical results that I've just shown in phase 1, they had also good results with 10 micrograms. So the companies have used a dose of mRNA which is slightly higher than what was actually required. CureVac has used a dose which is much lower, and we hope that it will be enough to vaccinate and to get a neutralizing effect also in elderly patients. But I would say that at the end, the technologies are probably similar, and the companies could overall have probably used a similar dose. There are small differences in the liposome. So the liposome from Moderna, BioNTech, and CureVac are different. So it could be that the one of BioNTech or of CureVac is better than the one of Moderna. So there might be small differences, but in general, I think one should see these doses as have been selected, but they could have been lower or higher. So at the end, it's more or less the same range, I would say.
Okay, many thanks. The next question is, which cells are typically targeted by mRNA vaccines after injection? And which cells are important for the immune response? In other words, how does the expressed membrane-bound antigen make it from the injection site to the lymph node?
This is a question I could not answer. So the injection intramuscular of this formulation is new. So whether the liposomes in the muscle transfect muscle fibers or fibroblasts or immune cells which are in the muscle, there are not many immune cells in the muscle. So that's a difficult question to answer. I would say that I would expect that mostly the liposome transfect muscle fibers and muscle cells, and that then the immune response is made by so-called cross-presentation. So where dendritic cells take up the protein, or maybe even take up the mRNA from several cells and present it. So I would assume that this intramuscular formulation of mRNA are proceeding by cross-presentation. It's not the case for the mRNA vaccines, which are in the clinical development for cancer patients. These vaccines are given intravenous, and they deliver the mRNA content to antigen-presenting cells in the lymph node and the spleen directly. So, but these formulations for cancer patients, again, are given intravenous. Here, the intramuscular injection, I would assume, is transfecting muscle cells, and cross-presentation allows the development of an immune response. But it's not yet demonstrated as well.
Many thanks. I think here you already have partially answered the next question, which is, why is it not feasible to use naked mRNA vaccine for COVID-19 as was done in the cancer naked mRNA vaccine study? And I think one answer is what you already gave, is the application form.
Yes, and the mRNA vaccine naked that we made, for example, in CureVac, use much higher dose of the mRNA to be efficacious. So here one has to keep in mind that the dose of 100 micrograms, 30 micrograms, or 12 micrograms are very low. In the early clinical studies, we use 800 micrograms per injection. In mice 20 years ago, we used a few hundred micrograms per mouse. So here one has to keep in mind that these liposomal formulations, they are very efficacious, so they allow to use much less mRNA. If we would have been intradermal with non-replicating mRNA, you would need much more mRNA to get a good immune response. So the liposomal formulation allows to go much lower with the dose and to get a high efficacy with a very low dose.
Many thanks. And the last question is, if these vaccines are quite easy to manufacture, why is it difficult, for example, to develop a new vaccine, for example, against hepatitis C?
I don't think it's difficult to develop a new vaccine against hepatitis C. So the development is not a problem. So probably it's the efficacy, the clinical studies. I'm not sure how one can organize clinical studies nowadays to make a prophylactic vaccine against hepatitis C. I think the problems are more in the design of clinical studies than in the production. The production of mRNA vaccine is easy.
Many thanks. I would like to say thank you also for the beautiful presentation. Also in the chat, there were numerous remarks. Many thanks for this very interesting and in-depth presentation. And with this, I would like to applaud also with the virtual clap. Many thanks for taking the time. And with this, I will step over and would like to introduce Florian Kramer. Florian Kramer received his advanced training in biotechnology and applied virology at the University in Vienna. He then did a postdoctoral work in the laboratory of Peter Palese at the Department of Microbiology at Mount Sinai, New York, and he is currently holding a position as a professor of vaccinology at the Department of Microbiology at the same institution at Mount Sinai in New York. He is the principal investigator of the Sinai Emory Multi-Institutional Collaborative Influenza Vaccine Innovation Center, and he is part of the NIH-funded Centers for Excellence in Influenza Research and Surveillance, focusing on understanding broadly reactive immune responses against the surface glycoproteins of RNA viruses. And that's one of the reasons why he is here, and I'm looking forward to your talk. I'll hand over, the Zoom is yours. Please, Florian.
Thanks a lot for this nice introduction. Yes, I have to admit I'm biased. I'm interested in antibodies and glycoproteins. So I hope you can see my presentation.
Yes, it's fine. You can start.
Great. So I'm trying to answer the question, how important are neutralizing antibodies in the management of COVID-19? Just before I start, I want to disclose my conflicts of interest. We are working here also on a COVID vaccine at Mount Sinai, and I have been named as inventor on IP for that and for serological assays for SARS coronavirus 2 serology. So before we really start to talk about what type of immune responses protect us from SARS coronavirus 2 infection and disease, we have to review the targets of the immune response. And so on the left side here, you have a virion of SARS-CoV-2 schematic. You're probably very familiar already with this schematic. We have the RNA on the inside of the virion that's covered by nucleoprotein. There is a lipid envelope with two proteins in the envelope protein and the matrix protein. And then we have the spike protein on the surface. And the spike protein, of course, is the protein that is used by the virus to bind to a receptor, to ACE2. And it does that with a little part of this relatively large spike protein. And this little part is called the receptor binding domain. And so, of course, if you have these proteins in the virion, the body is going to make immune responses against these proteins, and we know that the antibody response, for example, against the nucleoprotein and the spike protein is relatively strong. We shouldn't forget that this is a very large genome, and there's a lot of open reading frames on the genome. I'm showing this here on this schematic. So there's a lot more in terms of antigens than just what you find in the virus particle. So we have to keep that in mind. And so when we talk about virus infections and immunity, we talk about correlates of protection. So it's always very interesting to figure out, and very helpful to figure out what actually protects us from the infection. Some immune responses might be there, but they might not be useful. Some might even be detrimental, and other types of immune responses might be protective. And so I wanted to go through that in the beginning. I'll start here with the antibody responses. What do we know about antibody responses to SARS-CoV-2? We see antibodies to spike and or the RBD, and those are often neutralizing. It's important to keep in mind that there's also neutralizing antibody epitopes outside of the RBD. We know that there are antibodies to the nucleoprotein, to non-structural proteins like ORF8, and they are usually non-neutralizing. So far, we have seen that FC-FCR interactions, basically, the interactions between the FC part of the antibody and the effector cells seem to play not a really big role in protection against SARS-CoV-2. At least this is based on some animal studies. I think more work needs to be done. And what we also know is that we get regular IgM, IgA, and IgG responses. The IgM and IgA responses seem to be relatively short-lived, although there's also papers that describe a long-lived IgA response. And the IgG response seems to be like a normal IgG response to a respiratory virus. And of course, after natural infection, there's also mucosal antibody present. And this is something that we miss with the current vaccines. I said that these antibodies against the spike protein are often neutralizing, and this is shown here. On the x-axis, you see binding ELISA titers against the spike protein. On the y-axis, you see neutralization against activity against authentic SARS coronavirus 2. And there's really a nice correlation. I'm just showing this example from one of our papers, but many people, many investigators have shown that. So there's a really nice correlation between antibodies binding to the spike and antibody and neutralization of the virus. Just to show you that these antibody titers are relatively normal and relatively long-lived, here's also data from one of our studies. On the left, I'm showing you binding titers in 120 individuals over time. We bled them around day 30, around day 82, and around day 148, so 5 months out. And while there is some decline, it looks relatively normal. There's almost no decline between day 30 and day 82, and then there is some decline out to 5 months. We also have now data from 5 to 7 months, and it's basically a straight line. And the same happens with neutralization titers. So they go down to some degree, but they don't disappear. And in some cases, we even see increases. We haven't figured out yet if this was re-exposure. Or if this was if this is driven by affinity maturation. And 5 months out, if you look, there's still a nice correlation between binding antibody and neutralizing activity. but there's also other factors, other parts of the immune response that are important to look at. Memory B cells, for example, and this here is from a very nice paper by Alex Sette and Jane Crotty at the La Jolla Institute of Immunology. We know by now that very nice immune memory in terms of memory B cells is formed. This here is against the spike protein, and you can see that after infection, memory cell populations go up and then they stay constant. And memory B cells can be a correlate of protection, and in fact they are for hepatitis B, for example. They're very important for virus infections where you have a long incubation time. And then we have, of course, T cells. I'm not a T cell biologist or T cell immunologist, so I'm not going to go into detail here, but of course, it has been shown that there is a substantial T cell response to SARS-CoV-2 infection. It's more dominated by CD4 T cells, but there's also CD8 T cell response. There are a lot of these T cells that respond to the structural proteins, but there's also a response against these open reading frames that are not part of the virion. And again, this is from work that Jane Crotty and Alex Sette have published. Okay, so which of these immune responses do protect us? And of course, the first data that came out was from animal studies. What I'm showing you here are two papers from Dan Baruch's group. On the left side, you have an reinfection study in non-human primates. On the left side, you see what happens after the primary infection. This is our genome copy numbers in the bowel, which is basically viral titers. And you see that these animals get infected, and then at some point they clear the virus. And then when they tried to infect these animals at the later time point, they saw that these animals were protected from reinfection, most likely due to an adaptive immune response. There is a little blip here. This is probably input virus, but the protection was more solid in the lung than in the upper respiratory tract. That needs to be said. And at the same time, they did a study with an imperfect experimental vaccine against SARS-CoV-2 that gave a variety of neutralizing antibody titers. And then they looked at correlation between virus titers in the lung and neutralizing antibody titers. They found this very nice correlation between basically neutralizing antibodies and protection. The important part here is that the titer that was needed to give complete protection was relatively low. It was a neutralizing antibody titer of 1 to 100. By now, there's, of course, much more work. We know that monoclonal antibodies that neutralize can protect animals. And we also know that if you vaccinate non-human primates, transfer the serum into another set of non-human primates, you get protection. So antibody is sufficient in this model for protection. This was, by the way, also work that Dan Baruch published. And having antibody titers as correlate of protection is nothing new, right? So this is from a paper from a review from Stanley Plotkin. And this is a list of existing correlates of protection against all kinds of diseases. We have correlates of protection for hepatitis A and hepatitis B, which are basically ELISA titers. We have the famous HAI titer of 1 to 40 against influenza, which reduces the risk of getting infected by 50%. We have neutralizing activity in the case of measles. So we have all of this for many different infections. It's usually antibodies that correlate here with protection. That doesn't mean that the antibodies are the only thing that protect in this case, but they might indicate a protective immune response. And so it would be very good to also have that for SARS coronavirus 2, for basically public health reasons and also for patient management. And so in humans, the first evidence came actually from a really cool study that the Bloom and Graninger lab did in Seattle. They recruited the whole crew of a commercial fishing vessel, 122 individuals, into a study. They bled everybody, determined neutralizing antibody titers, and they found that 3 people had neutralizing antibodies. And then the ship left the harbor, and they had an outbreak with SARS-CoV-2 on the ship. And as it happens with outbreaks on ships, we know that from the cruise ship outbreaks from last year, there was a really high attack rate. 82.5% of the crew got infected, but the 3 people with neutralizing antibodies didn't. And that's a small number, but it's statistically significant. And now, of course, since then, there's a lot of data, and I'm going to walk you through that. There's a very nice study from the UK that was published in New England Journal of Medicine by Lumley et al., where they had recruited 12,000 healthcare workers, the majority of them serologically negative, but also about 1,300 serologically positive. And then they started to follow them for 6 months and did a PCR for virus every 2 weeks. And what their data showed is that basically 223 of the negatives had a positive PCR, and only 2 of the serologically positive individuals had a positive PCR, and those 2 were asymptomatic. And what they show is that basically you had a risk ratio of 0.11, meaning that there was significant, very strong protection by being seropositive, not just against symptomatic infection, no, against any type of infection. And that is really good, and that might be better than what we see even with the RNA vaccines. There's another study from the UK that's out as preprint, the Siren study, another very large and really excellent study, where again, healthcare workers were followed. They had 14,000 serologically negative ones and about 7,000 serologically positive ones. They also did PCR tests every 2 to 4 weeks, and they found 318 PCR positives in the negative group and 44 in the positive group, the antibody positive group. They also had, in addition, 94 people that seroconverted in the negative group but didn't have a positive PCR, probably because they missed the window. They didn't even put those into the analysis. But when they just did the analysis with the PCR positivity rates, they came to the conclusion that they had an 83% lower risk of infection in people who had antibodies. And again, this is against asymptomatic and symptomatic infections. So this is much more stringent than what we do in the vaccine trials. There's another study that basically shows the same, and this is from the US now, in about 3.2 million individuals, where they basically put together data from all the commercial testing facilities in the US, and they looked for people who became positive, serologically positive, and then they followed this group and looked how many of these people over the next period of time became PCR positive. And they compared it to people who were serologically negative. And what you can see here is that initially, many of the people who just became antibody positive are also PCR positive, which makes sense because we know that the RNA of the virus sticks around. But over time, the rate of PCR positivity in this group of serologically positive individuals declined and was just a small fraction of what you see in serologically negative individuals. So again, the same point, and again, this also includes asymptomatic infections. And the last study that I wanted to show you was done in recruits, in Army recruits, actually Marine, United States Marine Corps, in about 3,250 individuals. The new recruits came in, they were quarantined for 2 weeks to make sure that they're virus negative, and their antibody titers were assessed during that time. And then they went into training, and during training they were tested 3 times bi-weekly by PCR for presence of virus. And what they found was they had 189 seropositive participants, and 19 of those were at any point then PCR positive. And they had 2,247 seronegative ones. Of those, more than 1,000 became PCR positive. So the risk reduction here is a little bit, the reduction is a little bit lower here. It's only 5-fold, but these are very crowded quarters. As we know from 1918, in these army barracks, it's very easy to get infected. Nevertheless, there was a significant reduction, and they also looked at that by antibody titer. And there we see, and on the upper side, on the upper panel you have spike and the spike titers. On the lower panel you have RBT titers. You see that the ones that got infected had very low titers, and the higher the titer, the more solid was the protection. So just a little excursion here. We see that natural infection gives us significant protection from reinfection, and that's correlated with being seropositive. But how do we handle people who already had an infection? Should we vaccinate them? And if yes, how often? I'm bringing this up because there's a lot of new data coming out. I'm going to show you some data from our study, but there's in total 9 papers out right now that all show the same. So we have a cohort where we also look at correlates of protection, mostly healthcare workers, and a lot of them had infections in spring and now got vaccinated. While at the same time, we also have people who didn't have an infection, were seronegative and also got vaccinated. And we wanted to compare what happens after the first shot. And so this is what I show you on the left side. These are antibody titers. In blue, we have the people who were at baseline naive. And in orange, we have the people who were positive for SARS-CoV-2 by PCR or were positive by serology. And then they got their first shot of mRNA vaccine. And what we see is that the people with pre-existing immunity mount a very rapid antibody response, while the people who are naive have a very delayed response and it doesn't go up as high. And even after the second shot, the people who were naive don't reach the titers that we see in people who had pre-existing immunity to SARS-CoV-2. In fact, if you have pre-existing immunity, the second shot doesn't give you really a benefit in terms of antibody titers. We also looked, and previous speakers talked about specifically for RNA vaccination side effects that become more severe after the second shot. We showed in our paper that this is actually, in a way, also true for people who already had SARS-CoV-2 infections. Just in their case, the side effects are already way worse than for naive individuals after the first shot. While after the second shot, there's actually not much in terms of reactogenicity anymore. And this is shown here. It doesn't apply to the local side effects, but it certainly does apply to the systemic side effects. All right, and so to end, I also wanted to look at neutralizing antibodies as a correlate of protection after vaccination, and we assume that they are, but the truth is that the current data is messy, and we'll see how much the new data with the variants can actually help to reduce the messiness of that data. So just as a primer, and you have seen this this data set in the slides of the previous speaker, this here data from the Pfizer-BioNTech studies where people were given 2 different vaccines. Please just focus on the BNT162b2 here. This is here shown for 2 different age groups, 18 to 55 and 18 to 85. So this is the vaccine that was then licensed or authorized. And the vaccine was given on day 1 and day 21. And what you see, and you see that in all dosing groups, is that after the first vaccination, so meaning until day 21, there's very little neutralizing activity in these people's serum. Very few people have neutralizing activity. And even if they have neutralizing activity, it's relatively low. It's consistent across age groups and it's consistent across doses. However, when you look at the data from the phase 3 trials, and this is from Pfizer again, but Moderna shows exactly the same, you see that the protective effect already kicks in after the first dose. So all of you have seen this graph probably. On the x-axis, we have time after first vaccination. On the y-axis, we have incidence of cases. Red is here the placebo control group, and you see the cases climbing. And in blue, you have the cases in the vaccinated group. And you see that initially until around day 10, these 2 lines basically are very similar, and then they separate around day 10. and this is before the second vaccination is given. This is before these people induce a strong neutralizing antibody response, which they do after the second vaccination on day 21. And so, of course, that brings up a lot of questions about how much neutralizing antibody is actually needed and what other factors are protecting during that time frame when neutralizing antibody titers are low. And so that's why I said that the situation for vaccination is a little bit more messy. And so just to conclude, protection after natural infection is robust and as good or even better than after vaccination. And I'm completely aware that there are cases of reinfection, and there might be even cases where reinfection causes severe disease. But those are rare, and quantitatively, if you look at it, this statement is actually really true. The protection is correlated with antibody responses to spike. We have to be a little bit careful because the CD4 response to spike and spike antibodies also correlate with each other. So what we see here in terms of the antibodies to the spike could be in a way a surrogate for another immune response that we don't measure that might actually contribute to protection. We know, though, that therapeutic monoclonal antibodies work in humans. 2 are in use in the US, and they show efficacy. And we also know that antibodies are sufficient to protect in animal models. That's true for hamster models. That's true for non-human primate models. It's true for monoclonal antibodies, and it's also true for polyclonal serum. We assume that neutralizing antibodies are likely a correlate of protection for vaccines as well, but robust data is needed. And I think what we need to establish to understand and manage this disease better is a data that correlates with protection from disease, and probably a data that correlates with protection from infection. And so while these could be measured the same way, they are probably different amounts of total antibody. And it's very likely that much more antibody is needed to protect from infection, especially in the case of vaccines that are given intramuscularly that don't induce a lot of mucosal immune responses. And so in order to get a little bit of IgG onto the mucosal surfaces, you need relatively high serum antibody titers already, because that transfer is not very efficient. And with that, I would just like to acknowledge my team here at Mount Sinai and our collaborators at Mount Sinai and at other universities. Thanks a lot.
Thank you very much. Great talk, really enjoyed that. Now we just need to look at the questions. So, so someone has asked, should we do a PCR test prior to vaccination to prevent co-infection in case of asymptomatic infection? I'm not quite sure what that means, in fact.
Well, I don't think that is necessary. I mean, there's no co-infection in the way anyway, but I don't think that is really necessary.
How long do you think, do the studies give any indication as to the length of time immunity is likely to last?
Well, the problem is we haven't had enough time yet, right? So we need to do that in real time. But of course, the titers are initially, they are high and then they drop off, and that's true for vaccination, that's true for natural infection, right? And so the question is, when do you cross a titer that protects most people from disease? And that is really an open question right now. So what's really important right now is to have longitudinal cohorts, to follow them, to measure, to look at their antibody titers, to look at their T-cell responses, and see when infections start to occur. And a lot of these studies are ongoing right now. So they will produce results, but I think in order to figure that out, we just need time, right?
Yeah, sure. So someone's raised the question about whether any cross protection arises from previous infection with other endemic coronaviruses that circulate.
Yeah, that's a very interesting question, and we had a heated debate for the last 12 months about T-cell responses, right? And specifically, I guess, stockbroker and all kinds of people who wanted the economies to open up again were big fans of T cells, while most T cell biologists were very cautious about this. So there could certainly be some type of cross protection, specifically in terms of modulating disease severity. You know, it could be. We need relatively large studies to find that out. But there could be on the T cell side certainly some type of protection that is induced specifically by beta coronaviruses like HKU1 and OC43. If that protects you from an infection that's unlikely, but it might actually contribute to some people being asymptomatic and to having mild disease in others. On the antibody side, it's also an interesting question. We do see that if you get a SARS-CoV-2 infection, or if you get actually an mRNA vaccine with SARS-CoV-2, you do make a back boost against OC43 mostly, and also HKU1. There's no interaction, it seems, or no cross-reactivity to the alpha corona spikes from NL63 and 229E. We have isolated monoclonals that can cross-react, but most of them don't neutralize. And if they neutralize, and I think Scripps has 1 or 2 neutralizers, the IC50s are really high, meaning that the neutralizing activity is really low. So from the antibody perspective, I'm not so sure that there's much contribution in terms of cross-reactivity.
Okay, so there's quite a few questions here. So we'll go through a few of them and fairly quickly. Different people are asking in different ways. Do you need a second vaccine when if you've had the infection and you're given the first dose of the vaccine? Is there any need to give people the second dose of vaccine?
The US policy is you should get 2 doses. Many European countries have changed that policy, and there is 9 publications out there that basically say that the first one gives you very high titers and is beneficial. The second one is probably not needed, and the response is actually not very good. There's a very nice study from NYU, for example, that shows that the B-cell response against the second shot, if you already had COVID, got 1 vaccine, if you get the second shot, the B-cell response is actually almost non-existent and not very helpful. And so I think there is no need for giving those people a second vaccination. All that data is based on mRNA vaccines, but I can imagine that it looks similar for other vaccines. That data needs to be generated, but I wouldn't recommend giving the second shot. And I think the decision of countries like France to change their policy is really a smart one. You save also a lot of vaccine doses.
That's true. Although, of course, we haven't got the clinical outcome on that yet. But and then somebody has asked, what are the differences between naturally acquired and vaccine-derived responses, and which of those are important?
Yeah, that's hard to say. Okay. There are a lot of differences. Mucosal immunity is one, which is certainly present with natural infection, not so much with vaccination right now. We would need a mucosal vaccine for that. The responses against targets outside of spike are basically not present for any of the vaccines used in Europe or in the US. You might get some responses with inactivated vaccines. In terms of the importance, it's really hard to say. One thing that we recently found is that the ratio of neutralizing to non-neutralizing antibodies is better in natural infection as compared to vaccination. On the other side, the absolute titers with mRNA vaccination are usually higher than what you see even in people who had a natural infection and respond very well. So I'm not sure if that ratio is important at all, but it looks like the vaccination induces a higher number of non-neutralizing antibodies. But again, I don't know what's more important, the ratio or the absolute titers. And right now I would prefer a higher absolute titer. I'm not too worried about the ratio. So there are certainly differences, but we don't know exactly what they mean.
Thank you very much. Actually, there are a bunch of other questions, but we have run out of time. So I apologize to all those other people who've asked questions. We haven't got round to them, but there really are a lot. So I just would like to wrap up this session. Great meeting, I think. I really want to thank all the speakers for really excellent and stimulating talks, clearly stimulated a lot of questions. I really also would like to thank the audience for A, coming along, and B, asking plenty of questions. That's much appreciated. And it's been a great symposium, and I'm now going to draw it to a close. So thank you very much, everybody, and goodbye.
Goodbye. Thank you.
Thank you. Thank you. Goodbye.